In the best name of Allah Almighty, who is the most beneficent and the most merciful. I am Azharuddin from first year pharmacy. Today our topic of presentation is about calculation of the enzyme activity of catalase present in rabbit blood sample. First, it is important to know about the nature and the function of catalase enzyme. Catalase is an antioxidant enzyme found in all organisms using oxygen. It is present in sites of hydrogen peroxide production like peroxisome and mitochondria etc. In human, it is mostly found in liver. Function. It catalyzes the decomposition of hydrogen peroxide to oxygen and water. Low level of catalase in the body causes the condition which is called a catalasemia. If hydrogen peroxide is not decomposed to water and oxygen, it will be converted into reactive oxygen species, which is damaging to DNA, protein and cell membrane. So it's very important enzyme. Requirements. Hemolyzate e.g. 0.1 ml rabbit blood and 25 ml water. Substrate, which is 0.09% in phosphate buffer having pH of 7. Sulfuric acid, test tube, burette, conical flask, pipettes and water bath. Experiment. Take two test tubes. One is for the test denoted by T and another is for the control denoted by C. Now pipette out 0.2 ml substrate and 4.5 ml phosphate buffer and add it into test tube T and C. Next step is pre-incubation, which should be done at 40 degrees Celsius for 2 to 3 minutes. Add 0.5 ml hemolysate. Hemolysate should be added in test tube T, not to test tube C, because it is for the control. Incubate it at 40 degrees Celsius for 10 minutes. Catalase present in blood sample will convert hydrogen peroxide to water and oxygen, thereby decreasing its concentration. Now concentration of hydrogen peroxide in the test tube T will be less as compared to those in test tube C. Add 2 ml sulfuric acid and heat at 70 degrees Celsius for 2 to 3 minutes. Addition of sulfuric acid and high temperature would denature the enzyme effectively. Finally, titrate it against KMNO4.
Assalamu alaikum. My name is Misbah Latif and I am here to explain experiment number 11 procedure, that is to calculate the enzyme activity of catalase present in rabbit blood. So let's start with the procedure. So first of all, we have to collect blood samples from the marginal ear vein after removing the hair from the small area. Then 0.5 ml of blood, we have to collect it into a 3 ml capacity tube containing 45 USP units of lyophilized heparin. Basically, what is USP? USP is the unit of potency of drugs, and potency is an expression of the activity of drug in terms of the concentration or amount of the drug required to produce a definite effect. And lyophilized, it is also known as freeze drying. It is basically a process in which water is removed from a product after it is frozen and placed under the vacuum, allowing the ice to change directly from solid to vapor without passing through a liquid phase. Basically, it's a sublimation process. Then we have to mix the tubes gently so that there is a uniform mixing and to prevent the clotting of blood. Then a fresh phosphate buffer solution whose pH should be 7, containing 1% of hydrogen peroxide is prepared. Then we have to dilute the blood 10-fold with 2 to 9% solution of sodium citrate dihydrate. Then 25 microliter sample of this diluted blood, we have to place it in a boat floating on the top of 14 ml of the solution of hydrogen peroxide that had been prepared in a 50 ml Erlenmeyer flask with a side arm. So basically, an Erlenmeyer flask is also known as the conical flask or titration flask. It is basically used in laboratory for the titration purpose. Then each sample is represented by 1/400 ml of blood. Preliminary test indicates that this contains catalase activity measurable by the standard curve. Basically, a preliminary test, its purpose is basically used for testing of new drugs to determine the safety and efficiency of the potential substance among many other drug characteristics along the way. And standard curve is also known as the calibration curve. It is basically a type of graph that is used as quantitative risk. So now we have to calculate the activity of enzyme catalase present in rabbit blood by using the formula as given below. So for this we have to take 26.5 micromolecular concentration of H2O2. We have to take control, that is 0.3, and then we have to take the test, that is 0.2. Then we have to put all these values in the formula, and the final answer will be 132.5 micromole, and that is.
Assalamu alaikum. My name is Anusha Shamim and our group topic experiment is to calculate the enzyme activity of catalase present in rabbit blood sample, and I cover the results of enzyme activity of catalase present in rabbit blood sample. So सबसे पहले मैं ये बताना चाहूँगी कि enzyme होता क्या है. So basically enzymes are generally protein in nature. They act as catalyst. They speed up the rate of reaction. They have biosynthetic abilities, जिसका मतलब ये है कि वो organisms में बनते हैं. Now I tell you about catalase enzyme. Catalase enzyme is one of the fastest enzymes in the cells that used for detoxify या फिर remove करने के लिए यूज़ होता है किसको, हाइड्रोजन peroxide from the cell, जिसके नतीजे में water molecule बनता है और molecular oxygen भी बनता है, as you can see in this figure reaction. Now move towards the catalase test and its result. So after doing experiment in rabbit blood sample, if the catalase enzyme present in the sample of organism, visible bubbles product produced होने लगते हैं, but अगर catalase enzyme present नहीं है तो bubble observe नहीं होंगे, जिससे हमें ये results observe होते हैं कि the reading calculated above and its value is 132.5 micromole. High amount of hydrogen sulfate is used in the control and its value is greater than the test reading that is 0.3 and the test reading is 0.2. So the above experiment indicated that this contained catalase activity measured by standard curve. Also, catalase activity was higher in rabbit than in domestic animals and about half of the activity of human erythrocytes. Rabbit seminal plasma catalase is much higher than in the semen of other mammals. Further, I want to tell that the experiment shows that the female rabbits have lower hematocrit and catalase value than the males. Now, the disease related to catalase enzyme. जैसे कि हमें मालूम है कि कैटालेस कन्वर्ट करता है हाइड्रोजन पेरोक्साइड को, जो कि एक टॉक्सिक कंपाउंड है. सो दिस कैटालेस malfunction, this enzyme's malfunction leads to lot of multiple diseases like cardiovascular diseases, diabetes, hypertension, osteonecrosis. And basically osteonecrosis क्या होता है? It is the condition in which there is a loss of blood flow from to bone tissues, and also many disease caused by the low level of catalase enzyme, which is known as acatalasemia. Example: bipolar disorder, Alzheimer's disease, asthma, etc. Now I want to tell that biochemical importance of catalase enzyme. Catalase is one of the first in every realm of biological sciences. At the same time, it has a number of unusual features. The purpose of catalase in living cells is to protect them from oxidative damage, which can occur when cells or other molecules in the body come into contact with oxidative compounds, so they play an important role. फूड इंडस्ट्री में भी हेल्पफुल होता है कैटालेस एंजाइम, किस लिए? बेवरेजेस को बनाने के लिए और फूड की प्रिजर्वेशन के लिए. और टेक्सटाइल इंडस्ट्री में भी हेल्पफुल होता है. कैटालिस्ट इज़ यूज्ड टू रिमूव हाइड्रोजन पेरोक्साइड फ्रॉम द टेक्सटाइल फैब्रिक एंड दस इंश्योर दैट the finished products don't contain peroxide. It also helps in differentiating aerobic and obligate anaerobic bacteria. Thank you.